Bueno, Chengzhong, pero le dicen Michael. Se doctoró en China, en la Universidad Fudan, en 2002, publicó más de 400 artículos, tiene un índice H mayor que 100 y recibió varios premios y es editor del GMAT-ChemB y de Materials Advanced y tiene varias patentes y bueno, y nos va a hablar de nanoporosos y nanoestructurados, que es algo que nos gusta mucho en este campo, y está en la Universidad de Queensland, que no es la misma universidad de Adelaide. Please, okay, Michael, thank you.
It's so nice for me to visit Argentina, have the chance to tell you what I did before, and afterwards, most likely, I will tell you what I'm going to do in the following 5 years, something, in order to generate future potential collaboration. So that's the main motivation for this presentation. So before my talk, I'd like to introduce, to promote 2 journals of RSC. One is JMCP, the other is Advanced Materials Advanced, where I'm the associate editor. So JMCP is mainly about materials chemistry and the use of materials in biomedical applications, biology and medicine. And for materials advances, it covers all aspects of materials sciences. So if you are working in materials or materials for biological applications, you have some interesting work, feel free to submit your nice work to these 2 journals. I will be happy to handle your submission. Okay, so in my presentation today, basically I will talk about nanostructured and nanoporous materials. I will give a very brief introduction, then I will introduce mainly 2 parts. One is anisotropic nanostructured metal-organic framework, and the other is nanoporous materials in nanomedicine. This is mainly my research interest. And then I will tell you what I'm going to do in the future, as I said before, see whether we could find common interesting areas for our collaboration in the future. So why nanoporous materials? We know nanoporous materials include the micropore, small pore size smaller than 2, mesoporous materials, pore size between 2 and 15 nanometer, and the microporous materials larger than 50 nanometers. So there are different types of nanoporous materials, including, for example, zeolites with piercing walls, of course. This type of materials include morph and corf, and also nanoporous materials with amorphous walls, such as mesoporous materials like silica, carbon nanomaterials, etc. And also, it's a hot topic to have the morphology control of nanoporous materials, especially with the nanostructure or the morphology well defined. There's a reason for that. I will mention later on. So my background, my training background, actually I worked in the organic department of Fudan University, which is located at Shanghai, before I moved to Australia before. During my PhD, my training background is in mesoporous materials. So we know mesoporous materials, this is a development roadmap, and if you could control the pore size and the particle size very well, you can use these particles, we call them mesoporous, for example, silica nanoparticles. We can use them for a variety of applications, mainly due to their high specific surface area, tunable pore size, functionable surface area, surface chemistry, a lot of applications. Anyway, so I'm going to skip these details. when I talk about nanoporous materials with quilting walls, if most specifically metal organic frameworks, we know there are two pioneers in this area, Miyagi and Kitagawa from Japan. So ChatGPT always predicts these two guys may win the next Nobel Prize. Let's see whether this is true or not. So the first topic I'm going to mention is about nanostructured morph materials with anisotropy as catalysts. Why? So anisotropy means the property at different directions different. So we know for morph, due to the covalent bond, metal ligand covalent bond, there is intrinsic molecular level anisotropy. But if we can control the morphology very well, we know different facets may give you different properties. So this is called nanoscale anisotropy. If we could combine them together, for example, we can grow the second morph onto the first morph. We call this morph and morph. So if this is a morph and morph nanostructure, this would have the combined molecular and nanoscale anisotropy. And by doing that, the composition, facet, exposure, etc., all the anisotropy and indeed the property can be amplified. And these are not just the combination of two old materials. Indeed, by doing so, you can create a new interface. At the interface, you can create new active sites. And they should be considered as new catalysts. So that is our design principle using anisotropic nanostructured morph, especially morph and morph materials for different applications. So I'll give several examples showing that. Okay, so this is a work 5 years ago. You can see we can use the host morph and grow the guest morph either on the corner, on the top and the bottom surface, and you can do this 2 morph and 3 morph, all with well controlled morphology. And here is one example showing that we can control the second morph growth by changing the size. Actually, you are changing the interface coverage. And also, we have shown that the coverage is important in photocatalytic applications. So this is another example. Here, we can also show that by growing the second morph on the host morph, you can have unusual facet exposure of the second morph. So this is another example. I want to focus only on one application on hydrogen peroxide production. We know hydrogen peroxide is one of the 100 most important chemicals in this world. This is, I think, it's a multi-billion industry, and the demand is very huge. You can use hydrogen peroxide in many applications as energy source, in antibacterial applications, etc. So the current methods for hydrogen peroxide production mainly include 2 methods. One is an isochronyl method. However, this method may generate some environmental pollutants. The other is the direct synthesis through hydrogen and oxygen, but this process may have some potential risk concerns. So recently, there are quite popular 2 methods. One is electrochemical, the other is photochemical, but I'm going to only focus on electrochemical oxygen reduction to hydrogen peroxide application. Of course, this method is an alternative way for on-site generation of hydrogen peroxide and its use. Okay, so when we talk about high oxygen reduction reaction, usually you have 2 routes. One is the 2-electron ORR, which can generate hydrogen peroxide, but also you can have the 4-electron route, which can generate water. Of course, this is a side reaction we want to avoid. how to achieve only 2 electron ORR for high yield and high selective hydrogen peroxide reduction. Of course, we need the catalyst design. And also, for electrolyzer design, not only the catalyst, but also as Shi-Zhang mentioned before, we need to consider both the cathode and the anode. And also, you might want to consider the right solvent used to promote both the activity and the selectivity. So that's why I'm going to tell you what we can do to design systems, not just the catalyst, to improve the overall performance. So here I have several examples showing that increasing the anisotropy can improve the electrocatalytic performance. So this is one example showing that if you only have the cobalt or zinc-based ZIF material, they are not good catalysts. Only when you mix both zinc and the cobalt together, you can achieve a high performance. And moreover, you can see at the same zinc and cobalt ratio, you must have this 100 facet exposure, but not this 110 facet exposure. So this is another example. We could further use a ligand exchange protocol to further increase the anisotropy. By doing so, the yield rate can be increased by around 20%. This is another example showing that we can do some crystal engineering by making a big crystal into a small crystal. Then you're increasing the surface area, because mainly the external surface area is the active site for electrocatalytic reaction. And also this will also demonstrate that the increasing water content is also important in this electrocatalyst towards hydrogen peroxide direction. And also we have currently reached at NP level hydrogen peroxide production, reaching this value, and also with close to 98% percentage selectivity. So previously I mentioned the so-called anisotropic electrocatalyst design. So now I want to give you one example. That is the combination of the cathode and the anode design. So here we have replaced the anode OER to one so-called HMF OR reaction. So this is a 5-HMF oxidation into this monomer, which can be used for the production of biodegradable plastics. And this design can reduce overall voltage for this cell, and also the oxidation product value can be increased from water now to this monomer, and also the hydrogen peroxide performance can be improved. Of course, to achieve this design, we need the right catalyst. So here we use this conductive morph. This conductive morph consists of nickel as a metal center and HITP as a ligand. After we contract this conductive morph, we incorporate phosphotungstic acid into the pore channels. And by doing so, both the electronic structure of the photocatalyst can be adjusted. I'm not going into details, but here you can see the overall voltage can be decreased, either compared with the OER or the HER. So we can do this at a low voltage. And here you can see the hydrogen peroxide yield can be increased further, and the FDA selectivity is also very high. The mechanism is also demonstrated. okay, I can skip this part. Shi-Zhang mentioned one interesting work, that is use of seawater. Here I also mentioned one example using seawater for sustainable hydrogen peroxide production. So right now we only use fresh water and also electricity for this production. However, the seawater has a high amount. So what about we use seawater plus green energy such as from solar panel and for in situ utilization? So this is motivation. To do so, most MOF materials are not stable under seawater condition due to the high ionic stress and the corrosion problem. So here we demonstrate another catalyst, which is a metal phosphorus trichlorogenide with nanosheet morphology. The thickness is around 5 nanometer. What I want to highlight is that here we can, in this catalyst, each element, the metal, the phosphorus, and the sulfur, each element has a unique contribution towards the final performance. I will tell you in more detail. I will tell you more. Maybe I just use this slide to tell you. So what's the contribution of the sulfur? Because the sulfur can form 6 sulfur can form an octahedral cage, and the sulfur has a negative charge. So both the metal and the phosphorus are hidden inside this negatively charged cage. So this provides the intrinsic compulsion towards the chloride, as Shi-Zhang mentioned. In his case, it is hydroxide that repair the chloride, while in this case, it is a molecular structure that is octahedral sulfur cage that can do this job. Of course, we also do some deep engineering and identify nickel is the best metal center. And also the phosphorus can contribute to the adsorption of water and promote the hydrogenation process towards both thermodynamically and kinetically favored hydrogen peroxide production. There's, I think I don't have time to go to details here. So for details, we can go to this paper. So we can use this material in real seawater production for at least 100 hours. In simulated seawater, we can go to 1,000 hours. You can regenerate this hydrogen peroxide, use them for diode reduction, and use them for in situ generation for the application, for example, oxidation of glucose into formic acid, etc. So this is one example showing that we could use the well-designed electrocatalyst and then use seawater for the hydrogen peroxide production. And also we have demonstrated that this system can be coupled with a solar panel for the hydrogen peroxide production as well. I'm going to skip it here. So the MOF anisotropic design has a multiple advantage. So this is one example. We have an in situ conversion method. So we start from cobalt, zinc, ZIF-8 structure. Then have an in situ conversion of cobalt into one cobalt phosphorus active center, and then use this electrocatalyst for the nitrate reduction to ammonia. The advantage is that this ZIF-8 positively charged framework can provide an electrostatic interaction towards the nitrate negatively charged, and then discharge the positively charged product, which is ammonia. And also there are multiple active sites embedded inside this framework. So that's why the conversion rate of this photocatalyst is very high. So this is another example showing the so-called morph and morph design, also for nitrate reduction to ammonia, showing that you can form the new interface. And this new interface provides active sites, and these active sites eventually can give you a very high nitrate to ammonia reduction. This is another good, this is not electrocatalysis, but photocatalysis, showing that the nanostructure is a very interesting role, not just looking good, but also they provide very nice functionality. Of course, the design is based on what we have developed before. Basically, we can grow. So actually, the ground is one morph, which is a titanium-based morph, can be used as a photocatalyst. And on this floor, we can grow a nano house, which consists of mainly the ZIF-8 framework. And within this house, you can grow cobalt hydroxide as an electrocatalyst. As I mentioned before, this nano house will provide the driving force to absorb nitrate inside. And then this morph and morph design is indeed one S scheme heterojunction, which can provide some built-in electric field, then good for the separation of charge carriers. And eventually, this performance is very good. Basically, I can skip these details, showing that indeed there's adsorption, in situ adsorption, and a good performance. Anyway, here are several other examples showing that this morph and morph design is a very good strategy to generate new interface and new active sites, and eventually to promote the photocatalytic reaction. Okay, I think I can skip this detail. So here, what I have demonstrated is that the morph materials are very good catalysts. If you build based on existing, for example, stable and easy to prepare morph catalyst, but if you can build, as I demonstrated here, morph and morph catalysts, then you could generate new electrocatalysts with new properties. Okay, so let me change the topic a little bit. That is nanostructured porous materials in medicine applications. So, so far my group has developed many strategies to prepare silica-based porous nanoparticles with different topology and composition, etc., for biological applications. I think that the key is that we have we have this understanding of the correlation between the composition, pore size, surface topography, particle symmetry, etc., on their interaction with cells. For example, the adjacency, cellular functions, stability, degradation, adhesion, and cellular uptake, which give us some knowledge to use these particles for more delivery applications. I want to highlight one type of silicon nanoparticles, which is so-called nanoparticles with rough morphology. So our first work on this material was established more than 10 years ago. This is the first example using nanoparticles with rough morphology for cellular delivery applications. Afterwards, we have used this design for siRNA, DNA, mRNA, pesticide, cancer therapy, and even right now cancer immunotherapy applications. The advantage of this material is that they have rough surface. They can adhere to a variety of substrates. For example, in this case, the column, which would give you the opportunity for very prolonged release, so that, for example, you can have a tablet. You eat one tablet, maybe it can last for 3 days. This is one project ongoing in my lab right now. So why we want to prepare nanoparticles with a rough surface? This is because in nature you have a lot of similar morphology. For example, pollen grains can easily attach to the hairy leg of bees for easy pollination. And for virus, they also have this rough surface for the easy entry into the cells. So that's why, I mean, the infection capability of virus is so high. There are some detailed studies showing that these rough morphologies at a given density can form multiple entry claw, so to facilitate the endocytosis. So this is the first example, as I mentioned before. We can use a big particle and a small particle. One is negatively charged, the other is positively charged. Mix them together, spontaneously this particle can be formed. We have demonstrated that this rough morphology can increase adsorption towards a target molecule, slow release, and also the cellular uptake can be increased, so that for example, in this case, the gene silencing performance can be improved. The previous one, we used particles and particles with different size for the preparation. Afterwards, we have developed a molecular assembly strategy. For example, we only use TRs and some polymer precursors, using the TRs and the polymer self-assembly to get this silica nanoparticles with rough morphology. I have to mention that right now the nanotopography is different. In this case, we have this finger-like nanotopography. Previously, we have this semi-spherical-like nanotopography. We have some work showing the nanotopography difference is also important in terms of their interaction with cells. We have to follow this track and using different polymers to assemble these particles. For example, we can prepare silicon nanoparticles with rough surface with quite a uniform and controllable structure. What is the unique property of such particles with rough nanotopography? I want to use this slide to highlight you one interesting feature. So we know for conventional, for example, mesoporous materials, you can prepare very large pore. But for conventional mesoporous materials, for each pore, they are isolated. The space is isolated. But in our case, when you have this projection, but this space is indeed continuous. When we are using these nanoparticles for the encapsulation of linear structure, such as DNA and mRNA, think about this. This continuous space is good for the encapsulation of this linear structure. However, if you are going to fold DNA into small size, for example, 20 nanometer, it's going to be very hard. Indeed, the DNA backbone is very stiff. So that's why we have to use this strategy using this nanotopography to have a good encapsulation of DNA. And also, if we can control the spacing between the spikes, when this distance is small enough that it can provide a steric hindrance effect against some enzymes, so the stability can be improved. So we use this design principle for DNA transduction. So the model protein, the DNA express green EGFP. So if you can see green color, which means the DNA has been transfected to a protein with a green fluorescence. So you can see here our nanoparticle with this nanotopography is better compared to conventional silicon nanoparticles with large pores, of course, compared to the non-porous one. And comparable to the commercial gold standard. However, when you add an enzyme that can digest the DNA, you can see this lipofactamine, the transfection efficiency is dramatically decreased because they cannot, the DNA can be digested, but our particles can provide this protection against the DNAs that can digest the DNA. Okay, so this technology has been licensed to one company to use these particles, for example, for oral DNA delivery, etc. Okay, so previously when I mentioned the silicon nanoparticles with rough nanotopography, they are amorphous structure. So in our mesoporous material society, we know there's an innovation from a Japanese group. They can use bridged organoprecursor. They can make mesoporous materials with squeezing wall, right? So here we use that concept, but use this concept in the preparation of silicon nanoparticles, but with this squeezing nanotopography. So here you can see this spike. So this is a spike, but within each spike, you see this squeezing structure composed of the pi-pi conjugation of benzene ring. What is the advantage of this one? You can see these particles in around half an hour, they can enter into the cells. The cell wall is damaged, releasing the cytosol, then the cell is dead. We can use this concept for cancer treatment. Why is that? This is because once you have multiple spikes with this benzene structure, you can see they can extract the phospholipid that is a major component within the cell membrane due to this strong hydrophobic interaction. And also we have demonstrated this spatial arrangement cause a large area of damage towards the cell wall. Of course, you can use this concept for the treatment of cancer, for even the performance of this particle itself is not that good enough. But think about this, we always consider silicon nanoparticles have good biocompatibility. But once you add the composition control, now we can have this anti-tumor performance. You can also do some silica symmetry design from this symmetrical structure to asymmetrical structure, showing some performance, etc. Now I want to tell you some more biological related project. Here is one motivation. You know, in the tumor microenvironment, there's a rich accumulation of lactate, which is responsible for the immunosuppressive. The concentration is so high, there's no method that can directly deplete the lactate. So what we have developed is by inject the lactase into the tumor microenvironment. And by doing so, you can generate hydrogen peroxide, causing, and also we add, it can increase the hypoxia, etc., and eventually use this in cancer therapy. But in order to achieve this, you can see here, the lactate concentration is close to 30 millimolar. By our method, you can see the lactate concentration can be significantly decreased from 30 to 0.1. So that's a significant change. However, think about this. You cannot directly inject the lactase. Otherwise, you will generate a high concentration of hydrogen peroxide that can directly kill the rat used in our animal test. So that's why you do need a nanocarrier that can immobilize this enzyme with very high capacity and also with a slow release mechanism for this lactate depletion. That is the key in this design. Silicon nanoparticles can be used for enzyme delivery. Of course, then you can use this concept in a lot of other examples. This is one design showing that we can use this for the delivery of hyaluronic acid that can in situ generate this guy inside this joint. This is good for arthritis treatment. Just one concept. You can also use these silicon nanoparticles, etc., for DNA delivery I mentioned before, and also for mRNA delivery. For mRNA delivery, the key in our design is that we use these nanoparticles designed not just as a carrier, but also they can provide additional mRNA translation function. That is the key. For example, we have identified the higher GSH level is responsible for the mRNA decay, especially in cell lines with high GSH levels such as macrophage. That's why macrophages are always considered as hard to transfect cells. With this understanding, you can incorporate one composition in the nanocarriers that can deplete the GSH. For example, you can add the disulfide bond or tetrasulfide bond, and by doing so, both the mRNA decay can be inhibited. Moreover, this product, GSSH, can also enhance the mRNA translation due to the increased level of MTYC1. So that is one major finding in our case. We can also use this concept not only for cell type specific delivery, but also for cell phenotype specific delivery. For macrophage, we know there are mainly two phenotypes. One is M1, the other is M2. I might mention that a little bit in details later on. So we understand right now the M2. Before I mentioned macrophages are hard to transfect, but M2 is even harder to transfect compared to M1. With this understanding, you can use strategies to convert the M2 phenotype into M1. Then the gene delivery efficiency can be further improved. So this is work we have just finished. I think I can skip this one, too heavy information here. After we have done some cellular delivery use nanoparticles, so the recent years we have paid a lot of attention to the tumor microenvironment. This is when we talk about the tumor treatment. Why tumor treatment is so difficult? This is because in tumor you have not just tumor cells, but a lot of other immune cells. Understanding nanoparticle interaction with these different cell lines and the strategies that can combine the impact towards different cell lines, that would be the most important thing for tumor treatment. So far we have done the development of various nanostructured adjuvants. Adjuvants are useful components to boost the immune response, especially in vaccine formulations. So we have several generations of nanostructured adjuvants, including cattle trial completed in collaboration with African scientists. We have used the so-called bridged organosilica as immune adjuvants. This would give us the opportunity to understand not only the nanostructure, for example, the particle size, the pore size, but also the chemistry. That is the bridge group. For example, this is ethylene group, the phenol group, the biphenyl group, tetrasulfur group, etc. on the impact of these adjuvants and the adjuvancity when used for vaccine formulations. And we can also do some interesting fundamental chemistry here. So we know aluminum silicates, the major composition for zeolite. Here, what we have done is that we use this concept in the design of aluminum silica nanoparticles. But we have done is that we can control the coordination chemistry of this aluminum either in this 6 coordinate or the 4 coordinate. And we can see these two adjuvants, the 6 coordinate and the 4 coordinate, they have very different property, adjuvancity. For example, here, we could introduce binuclear 6 coordinate aluminum onto silica surface. And because of this rich hydroxyl group, it can provide the binding towards the protons. Then it can induce enhance the cellular when it interacts with proton, it will generate a positive charge. This will increase cellular uptake, and also the binding towards protons will give you this endosome escape performance, which is important for some cellular immunity. So that is the role of 6 coordinate. And also, if we end up with 4 coordinate, you need a cation to balance the negatively charged. In this case, we have the sodium. When these particles enter the cells, indeed, you have a high concentration of sodium, then this will end up with an efflux of potassium and then trigger a range of immune response afterwards. Eventually, they can be used for apoptosis-based cancer immunotherapy. For cancer cells, you need to amplify the immunogenicity, which is called the ICD. And here we can use silicon nanoparticles to amplify the ICD to make the cells from immunotolerant to immunogenic. So this is one example. That is a modification of cobalt and organosilica particles. And this, I want to highlight one recent work. Here we can use the cancer cell. Here is about we have different strategies for cancer cell to increase ICD. Okay, so this is to reduce one suppressive signal, which is PS, and also we increase PAM expression and decrease PS, and then we use the metabolic regulation to amplify the ICD. This is another strategy we can achieve. What I mentioned in the last slide is about the cancer cell. But as I mentioned before, in the tumor, there are also a lot of immune cells, such as the macrophage. Macrophage can account for around 40 to 50% of the cell number in the tumor. And the M1 type is mainly for tumor growth. The M2 is for tumor growth, and the M1 is for anti-tumor. So we have strategies to polarize the M2 to M1, such as using iron oxide to trigger multiple pathways. And also, we can use metabolic regulation. This is because for the M2, it's mainly about oxidative phosphorylation, and for M1, it is glycolysis. We have a strategy to promote obstacles to glycolysis so that we can achieve this phenotype change. I must mention that this strategy is very different compared to the cancer cell. In cancer cell, this metabolic pattern is different. Okay, so I can skip this one. So possibly the last slide. What I want to mention is that the cancer cell metabolism and the macrophage metabolism are on the reverse trend, especially for cancer immunotherapy. Then, is there any way that we can combine this metabolic regulation at the same time? For example, for the macrophage, as I mentioned before, you want to use the you want to increase the glycolysis, but for the cancer cells, you want to reduce the glycolysis. How to achieve this? Seems like a mission impossible. However, we have identified one intrinsic molecule for this immune modulation, which is hydrogen sulfide. So we could use hydrogen sulfide downregulation to promote this glycolysis upregulation and this glycolysis downregulation in the cancer cell, and this is in the macrophage. To achieve so, this is also based on spiky nanoparticle, which I'm quite familiar with. So we use a bimetallic morph, which is zinc and iron. And under mild acidic conditions, this morph can in situ generate iron oxygen hydroxide. This compound is responsible for this hydrogen sulfide depletion, which is already used in industry. And by doing so, we can achieve simultaneously metabolic regulation towards both macrophage and the cancer cell towards cancer immunotherapy. And also, I think I can give, so as I mentioned before, this is what I've presented is almost what we have already completed. But in the future, I think I will continue work on the cancer nanoimmunotherapy project. And also, I'm also interested in oral delivery, gene delivery, and the cancer phenotype specific delivery, which I feel this could be the future direction, not just cancer type, not cell type, but also you need to consider different phenotype difference. Can we achieve specific delivery? And also, I'm working in some areas on nanotechnology for agriculture applications, because in Australia, the wool industry, the cattle industry is quite developed. That's why there are a lot of requirements where I think the nanotechnology can provide some suggestions. For example, we can use the nature compounds without causing toxic effect to environment, to human, but for some applications in cattle, in sheep, and some catalytic work, as I mentioned in the very beginning. So this is about cancer immunotherapy. Can we use the right technology to further improve the survival percentage for cancer patients, especially in combination with the current cancer immunotherapy technologies? I think there's still a lot of room for our nanotechnologists working in this space. So this is what I mentioned. Can we develop nanotechnology shifting from cell type towards cell phenotype specific delivery. There's a lot of room to play right now. And there are some room for our nanotechnology for antibacterial application, parasite application, for antifungal application as well. This is what I mentioned for the current sheep and cattle industry. There are a lot of problems, and there are very few solutions. For example, in Australia, the common practice is you have to cut the tail, but in this technology will be banned very soon. How we could use technology to assist this, and I can skip this battery one. So basically, this is a summary of what I have mentioned. My group is mainly working on the nanostructured and the nanoporous materials for healthcare and the sustainability applications. So I think the main motivation for this presentation is to tell you what I did before, what I'm going to do next, and hopefully this could be an opportunity for us to generate mutual benefit collaboration in the future. Okay, thank you very much.
Thank you very much for your nice presentation. I think, questions?
Yeah, I have a couple of questions. At the beginning, you talk of hydrogen peroxide production. Now, it's very well described from the 60s that hydrogen peroxide can be produced by reduction of oxygen on carbon in alkaline solutions, the work of Jaeger, on even modifying the carbon with anthraquinone, the work of Shi-Zhang. Now, the question is, what is the benefit of going to a more complex system like a MOF in terms of stability, cost, as compared to a simple carbon electrode? This is the first question. And the second question is about the lactate peroxidase, oxidase. If you encapsulate the enzyme to reach the tumor and reduce in a controlled way the lactate, you also need oxygen. So maybe the oxygen concentration, the local oxygen concentration, will limit your technique. Can you address that?
Yeah, you're right. Let me answer your second question first. We haven't measured whether the hypoxic condition would affect the lactate depletion efficiency or not. But as I have demonstrated in the results, it is the depletion efficiency is already high enough. So that's why we haven't considered the hypoxic condition. If you see the results from 30 to 0.1, we feel that efficiency is already good enough. That is mainly because the lactate, what we have measured is in situ depletion, which takes several weeks already. So this is not a reaction in the glass tube something. So I think that oxygen level over a duration of several weeks should be already high enough for the lactate depletion. Okay. For your first question, I think that is a very interesting one. It really requires the whole community to think about this, and eventually what I have known is that there are several pilot trials across different countries using electrocatalytic production for hydrogen oxide. The advantage for me is that, in my mind, the plant using isoclinal process is huge. It's huge. Even the cost, because it's well developed. But this strategy produces hydrogen peroxide. You need to deliver, you need to transport this to different areas, right? But in, for example, let me give you one scenario. If in Australia coastline, we have a lot of agriculture applications where you need antibacterial agents. For my application, actually this is my ongoing project. There are common practices right now using hydrogen peroxide plus sodium chloride as antibacterial, antifungal treatment options. In my case, we simply use, this is already demonstrated in my paper. When we have sodium chloride in seawater, and we can convert seawater into hydrogen peroxide, this is exactly the same composition used in the current treatment. So I'm not saying this process will replace cranial process in a short term, but it may provide a lot of on-site applications, such as in some coastline scenario.
Another question?
Yeah, please.
Thank you for a very great presentation. I'm curious about your composites where you use the MOF and then you include, for example, a cobalt phosphide particle. How do you precisely locate these catalytic centers inside the MOF? Chemically? Do you use any specific confinement to synthesize them? Or?
You are talking about the active side, the active mechanism, or you are talking about the strategy to...
The strategy, yes, I was curious about the strategy to locate these catalytic centers, very small nanoparticles inside the larger cavities of the MOF.
Oh, okay. I think this is interesting. For the paper, I have mentioned the phosphotungsten acid inside. This is a good for me. I think this is a smart design, of course, by the student. The size is around 1 point something nanometer, and the phosphotungsten is a cluster, right? So this cluster size is a little bit smaller compared to the channel of this conductive MOF. Okay, so indeed it is triggered by the acid, and we can see using synchrotron, we can see there's a generation of this nickel oxygen phosphorus bond, and we have also used the in situ characterization together with DFT. We have identified both the phosphorus and the nickel contribute to the activity for the electron catalysis, and also on the cathode side and the anode side, the mechanism is a bit different. So I think, especially considering a MOF, you cannot use very big cluster. In this case, the match of the size of the guest and the host. So that would be.
I'm curious if you build the MOF around the catalyst, or you diffuse the
inside the channel. Exactly inside the channel. Inside the channel.
Okay. Like a ship in a bottle or something.
Yes.
That's interesting.
Yeah. You can consider the phosphotungsten acid as another ligand to replace the previous HIPP.
Okay.
Okay.
Another question?
Thank you for your talk, really nice. And a lot of information. So I was curious about the nanotopographic particles with the spike. So now you load DNA there, and you use it to delivery. So I have two questions. One is like how you load the DNA, if you use some strategies like charge or to increase the loading. And the other one is you say that the delivery was slow, or the slow release or slower release compared with other particles. So how you achieve that?
Okay.
Inside the particles.
Okay. So thank you for your question. The first question is that these nanoparticles are first phosphonate modified, which provides extra negative charge, and then conjugated with PEI. PEI is a polymer which is positively charged. that provides electron attraction force towards a negatively charged DNA. So the slow release mechanism may be related to the steric effect. So once you embed this linear structure inside this spiky surface, I think you may provide a multivalent attraction force, which causes the relatively slower release compared to, for example, on a plane surface. Okay, thank you. Yeah, please.
About the same nanoparticles, what is the size range of the spike and the total nanoparticles for the applications you mentioned?
Yeah, so we can do this around 100 to 500 range. So 100 to 500 nanometer in diameter. So we can have this level of control. For cellular delivery, I mean, it really depends. For example, the macrophages, they like to eat large ones. While for cancer cells, usually you need around 100 nanometer for the delivery application. And it really depends on which target you're going to use.
You have to optimize that.
We have all this optimization. There are some work showing the impact of the size as well. But I think this is well-known knowledge. The size of the nanoparticles should have a significant impact towards a different cellular delivery and in different type of cells as well.
Another question?
I have one question.
Yeah, please.
If the spikes, although they are covered with phosphor or phosphonates, are stable in the biological media or dissolve quickly?
We have measured the degradation profile. So if in you add in PBS, the degradation time would be in several days. So that's why I think this degradation behavior, I'm talking about pure amorphous silicon. So this would be similar to mesoporous material. But the thing is that you can control the diameter of the spike, both the length and diameter. So even we haven't done that, but I would assume the thicker the diameter, the more stable for degradation. So we should have some room. If you need a more stable one, we can make a larger diameter spike.
Okay.
Yeah.
And this spike help to enter into the cell to
Yeah.
cross?
Yeah.
You have measured that?
We have a lot of we have done
Okay.
a lot of this.
So another question? Well, we thank you very much.